Alright guys, welcome back to Underground Science, and in this video, we'll be talking about bystander activation, okay? So let's go and get started. So this is an important principle, right? It's an important principle in immunology. So the basic definition of bystander activation is this, right? So self-reactive T cells, self-reactive T cells, right? How are they self-reactive? They escape central tolerance in the thymus, right? Which we've already discussed in a previous video. So if they escaped the thymus, right, so they escape central tolerance, and then they unintentionally get activated during an immune response to infection or tissue damage. Key word here is unintentionally, okay? So a really basic summary of it is like, let's say you have a self-reactive T cell here, right? Let's say this is the self-reactive T cell, right? And there's some sort of inflammation, infection, or tissue damage going on here, and which is leading to a lot of inflammation, a lot of cytokine release. So what can happen is unintentionally these cytokines can stimulate and activate these self-reactive T cells, and these self-reactive T cells will then go destroy your own tissue. So that's bystander activation, right? The self-reactive T cell is a bystander. It's not involved in the actual infection and inflammation or tissue damage, but it gets involved because it's near this inflammation, infection, or tissue damage. And then once it gets involved, it gets activated and then destroys your own tissue because these are self-reactive T cells. Okay, so let's actually go through this and see how this works. So let's go step by step, all right? So first we need a trigger, right? Which is what? Infection, either bacterial, viral, or whatever, right? Just an infection. Or tissue damage, right? How do you get tissue damage and injury? Hypoxia, right? If you don't have oxygen going to a tissue, that can damage the tissue and lead to cell death and release of intracellular components. Or trauma, right? Can also lead to tissue damage. So if infection, right? Then remember, pathogens release what? Remember, pathogen-associated molecular patterns, right? And we gave specific examples in our previous videos. So like lipopolysaccharides, for example, and gram-negative bacteria are example of PAMPs, right? Flagellin is another example of PAMPs. Remember we said flagellin is released by many bacteria like Vibrio cholera, for example, or Helicobacter pylori, or Salmonella, or E. coli, or Listeria, right? And so on. And Pseudomonas, right? So those are PAMPs. Okay, like flagellin, like lipopolysaccharides, and so on, right? And that can activate, right, certain toll-like receptors, which we'll see in a bit. But if there's tissue damage, that will activate that. If infection, right, pathogens release PAMPs. I think I said activate PAMPs. That's not the right wording. It's release PAMPs, right? So if an infection happens, pathogens release PAMPs. And those PAMPs being lipopolysaccharide, flagellin, and so on, which can then, so PAMPs can go and activate toll-like receptors, right? But if there's tissue damage, you release DAMPs, right? Damage-associated molecular patterns. What are those, right? Well, if your tissue gets damaged, you release things like uric acid, like ATP, heat shock proteins, and so on. These molecules right here are called DAMPs, and DAMPs can also activate certain pattern recognition receptors, right, on your in your cells, for example, like your antigen presenting cells, okay, like NOD-like receptors, for example, right? So let's go and see how this works. So let's say we have an antigen presenting cell, right, in this case a macrophage, okay, and we have some sort of damaged tissue, right, whatever cause that may be. We have damaged tissue, and that's releasing certain self-antigens like uric acid, ATP, heat shock proteins, and so on, which are called what? Right? Damage-associated molecular patterns. So it's releasing DAMPs, right? And that will bind and stimulate certain pattern recognition receptors. And let's say simultaneously we have some sort of infection going on nearby too, that's leading to certain pathogen-associated molecular pattern release, like lipopolysaccharides, for example, that can also bind on toll-like receptors too, right? Like toll-like receptor 4. So binding of these two things, DAMPs and PAMPs, on certain pattern recognition receptors, like NOD-like receptors and Toll-like receptors, activates the antigen-presenting cell, in this case the macrophage. So when the macrophage then gets activated, right, it does a few things. It first starts releasing cytokines, right? A bunch of cytokines like interleukin 1, interleukin 6, TNF alpha, even interferons, right? So let's write that down. Even cytokines like interferons, right? Like interferons alpha and beta. Even like interleukin 2, interleukin 7, interleukin 15, right? Many different cytokines, okay? And so that happens. Another thing that happens as when these macrophages get activated, right, is that it starts expressing B7. Remember, B7 is really important because B7 needs to interact with CD28 of a T cell so that T cells can also get activated, right? So, but we got a little ahead of ourselves, right? We forgot to mention the fact that once these DAMPs and PAMPs bind, right, onto the pattern recognition receptors like Toll-like receptors or NOD-like receptors in our macrophages, then the macrophage gets activated. But remember also, macrophages ingest, right, phagocytose these antigens, whether that be self-antigens or pathogen antigens. So let's say it phagocytoses the antigens, right, and it presents the self-antigen, right, on MHC1, and it presents the pathogen antigen on MHC2, okay? So we'll follow that. We'll track this and follow this along, okay? So that happened, right? And then cytokines get released, and then we said another part of the activation for macrophages is the expression of B7, which then binds onto T cells, right? Specifically CD28 on T cells, right? And also remember the other factor in activation of T cells is the TCR and MHC interaction, right? So if we just focus on this one, right, this interaction right here. So remember we said, what is this, right? We said purple was pathogen antigen. So the pathogen antigen, so the macrophage, it phagocytosed the pathogen, broke it up, and presented it on MHC2, which we're already familiar with. So, and then what happens is a T cell can come by, interact with it, with its TCR, T cell receptor, and CD4, right? Remember, also binds MHC2. And then we get, remember, B7, CD28 interaction. That leads to the activation of the T cells. In addition, remember, the antigen-presenting cells releases certain cytokines. So after all these interactions happen, the macrophage can release interleukin 4, right? For example, and what does that do? It helps this naive CD4 cell to differentiate into Th2 helper cells. Okay? And remember, Th2 helper cells can release IL-4 and IL-5 in order to activate B cells and make them class switch and produce antibodies. Or what can happen is, after these interactions occur, right, the macrophage can release interleukin 12, and interleukin 12 can stimulate the CD4 T cell to differentiate into Th1 helper subtype. And we know Th1 cells do what? They release interleukin 2, which is important for CD8 T cell activation, so activation of cytotoxic T cells, and Th1 cells release interferon gamma, which then are important for attracting and activating macrophages. Okay? So that's happening due to this interaction, right? All of this is happening due to the interaction between the pathogen antigen, right, and MHC2 and TCR. But that's just a normal reaction, right? That's a normal part of activating our T cells, as we're familiar with, right? But something else that can happen is when we ingest self-antigens, for example, usually if you're ingesting something, right? So let's talk about this, right? So usually if you have an antigen-presenting cell and you're ingesting a pathogen from the outside, so an exogenous antigen or exogenous pathogen, it's going to phagocytose it, right? And break it up and present it on MHC2. Right? That's what happens normally. But what can also happen, right? And this is whether or not this is a pathogen or if it's a self-antigen, right? Or self-antigen. Either way, it can ingest it and break it up and present it on MHC2. But what can also happen is, let's say you have a self-antigen here, right? Or a pathogen antigen, like a viral pathogen, for example, right? This is a viral pathogen. Or this is a self antigen also, right? Because of tissue trauma, for example. Your antigen presenting cells can, of course, phagocytose this and present it on MHC2. It can phagocytose our self antigens also, present on MHC2 like we just talked about. But also what it can do, it can phagocytose your self antigens, right? Remember, once you phagocytose anything out, like if these antigen presenting cells phagocytose anything out here, you take it inside a phagosome, right, which is an endosome. So then you would have your phagosome, let's say here, right? And then with your self antigen, so we'll do the self antigen in yellow, right, in here. What can happen is something called cross-presentation. So instead of the normal expected output, where you phagocytose something outside, whether that's self or viral or some pathogen antigen, and present it on MHC2, what can happen is you can phagocytose something outside, right, exogenous, like a self antigen in this example, but instead of presenting on MHC2, what can happen is this self antigen can escape the endosome and go to the cytoplasm. Then once in the cytoplasm, right, you can have proteasomes that will degrade the self antigen into smaller peptides, and then we can present that on MHC1. So that's cross-presentation, right? This is cross-presentation. It's not, you're not presenting it on MHC2, you're presenting on MHC1. So that means we have four options, right? This antigen presenting cell can take the pathogen inside and present it, break it up, present it on MHC2. This antigen presenting cell can take the self antigen inside, break it up, present it on MHC2. Or it can take the pathogen inside, break it up, pathogen inside, right? Then it can escape the endosome, right? And then undergo cross-presentation, and you can present certain viral fragments on MHC1. Or you can take the self-antigen inside, right? And it can escape your endosome, then your proteasomes break it up, and you present that on MHC1. So now, if we take this cross-presentation pathway, right, you have, so let's clean this up, right? So we have our macrophage, or antigen presenting cell, and let's say we took the self-antigen, right? We phagocytosed it, it escaped the endosome, right, and went to the cytoplasm. So it was in the endosome, escaped, went to the cytoplasm, proteasomes broke it down, it went to the ER, right, the endoplasmic reticulum, got put on MHC1, and then you present that on the cell surface. It's your MHC1. And now this is your self-antigen, right? Due to cross-presentation, you have it on MHC1, not MHC2. Okay? So that's what I was trying to explain, is that you can also present things on MHC1 also. Like if you, for example, if you phagocytose something exogenous in the exogenous environment, you don't have to only present on MHC2. Your white blood cells, like antigen presenting cells, for example, can also present that on MHC1 also. Okay? So let's go back and see how this. So we already went through this, right? This is a normal antigen presenting cell T-cell interaction that we're all familiar with, right? But remember, cross-presentation, you can take exogenous self-antigens due to tissue trauma, right? Or some sort of pathogen infecting your cells, like viral pathogens, for example, can infect your cells get destroyed, release damage-associated molecular patterns like uric acid, heat shock proteins, ATP, which is represented in orange, right? These are all self-antigens. So what did this antigen-presenting cell do? It took those self-antigens inside. Instead of presenting on MHC2, right, a possibility was it escaped the endosome or the phagosome, got broken down by proteasomes, and presented on MHC1. So now this is a self-antigen, right? So if we have self-reactive cytotoxic T cells, because who interacts with MHC1? CD8 cells, right? Which are cytotoxic T cells. If you have self-reactive CD8 T cells that escaped central tolerance in your thymus gland, then it's going to bind to the self-antigen. And then, of course, the normal processes occur, right? So we know CD8 needs to bind to the MHC1, and then CD28 needs to bind to B7, right? And then you activate the self-reactive T cell. And of course, B7 is presented because of all the tissue trauma and inflammation that's occurring, right? So in all of these, this is showing that how CD8, self-reactive CD8 cells can get activated, right? But we skipped one person, right? Look at this dude right here, which I'll basically circle in white, right? In all this process that's occurring, right? This whole process, we're generating a lot of cytokines, okay? And just overall inflammation is happening. Now, all of these cytokines and inflammation in the process, what if there's a self-reactive T cell nearby, right? What if there's a self-reactive T cell nearby? So what do we see here, right? The self-reactive T cell, right, didn't need its CD28, didn't need to bind B7 or anything like that, didn't even need to interact with an antigen-presenting cell. Just being nearby, or the bystander, right, all of these cytokines, inflammation, can stimulate and activate your self-reactive T cells, whether that's a self-reactive CD8 T cell or self-reactive CD4 T cell nearby, okay? So, and then of course, if the self-reactive CD8 T cell gets activated, right, and let's say it's supposed to target any tissue, right, of the body, it can go ahead and do that and destroy our own tissue, okay? And same thing here, like this example is showing what happens if your self-reactive T cells interact with antigen-presenting cells, right? So as this process we discussed, right, the self-antigen interacts with a self-reactive CD8 T cell. This CD8 T cell gets activated, self-reactive, right? And then it can go to damage your self-tissue, okay? And so that's basically how it works, right? So you can basically have, like, if this was a self-reactive CD4 cell, right? Let's say this was a self-reactive CD4 T cell, and it got activated, right? And then the macrophage released certain cytokines like interleukin 4 or interleukin 12, right? If it released interleukin 12, we would have self-reactive Th1 cells, because we know interleukin 12 helps naive CD4 cells differentiate into Th1 cells, right? So then we would have self-reactive Th1 cells, and that would secrete a lot of interleukin 2 and interferon gamma, and that would bring a lot of macrophages and increase inflammation and destruction of your own tissues, right? So I hope that process makes sense, because at the end of the day, right, when we're like knowing all these processes, it's important to know like why even learn this, right? And what's cool is everything we just talked about, we can apply to real life. Okay, so if we go ahead and talk about type 1 diabetes mellitus, right? So let's see how that whole process, right? Why we even talked about that? Because we can apply it to a clinical scenario, right? Like type 1 diabetes mellitus. So let's say we have Coxsackie virus, right? And it infects your beta cells of your pancreas. So it infects your beta cells of your pancreas. Those beta cells get destroyed, right? Due to the infection of Coxsackie virus. Then when your beta cells get destroyed, it releases viral antigens, those of Coxsackie virus, and self-antigens, those of the beta cells. Like what? Like insulin, for example, right? So then, right, macrophages come in and engulf these viral antigens or self-antigens. Remember, we talked about cross-presentation, which will come into play here. So remember, we're representing viral antigens in orange and self-antigens in yellow. So let's take our pointer out. So our macrophages basically engulfed and phagocytosed all of these antigens, broke it down, and presented it on MHC2 and 1. So remember, it has options. Macrophages can take these exogenous antigens, engulf it, break it down, present on MHC2, or it can take these exogenous antigens, then these antigens can escape the phagosome and then get broken down by proteasomes, right? Then those peptides, right, can get put on MHC1s and then expressed on the cell surface. That's cross-presentation. So if, let's say, right, these self-antigens were taken in, but those self-antigens escaped the phagosome, proteasomes broke it down, and we presented those self-antigen peptides on our MHC1. And then also, let's say the macrophages, right, not only did they engulf self-antigens, there was also viral antigens here, right, of Coxsackie virus. So let's say our macrophages took in these antigens, our viral antigens, broke that down, but this time, remember when you're presenting MHC2, you take it into a phagosome, and then the phagosome fuses with the lysosome. You break that down, so you break the viral proteins down into smaller peptides, and you present that on MHC2. So we have viral antigens presented at MHC2 and self-antigens presented at MHC1. Now, one thing to know here, right, is that we can also have MHC1 that's presenting viral antigens, okay? We can also have MHC1, doesn't only have to be these options that we discussed, right? MHC1 somewhere else on the cell surface of the macrophage could be presenting viral antigens, right? And how is this happening? Same thing, right? If you engulf viral antigens, those viral antigens, right, intracellular viral antigens can get broken down by proteasomes in the cytoplasm, right, if it escapes the endosome or the phagosome, and then you can present on MHC1, right? Or the MHC2 on another area, right? Because these antigen-presenting cells don't just have like one or two major histocompatibility complexes, right? They have many, okay? So they have numerous MHC1s and 2s on their cell surface. So you can have your other option, right? You can even have, let's say this is MHC2, doesn't have to be presenting viral antigens, right? You could have engulfed self-antigens, broke that down via formation of the phagolysosome, and then presented self-antigens on MHC2 too, right? So maybe on another area of the cell surface of the macrophage, you have MHC2 that's presenting self-antigens. So let's talk about each case, right? So if we have MHC1 presenting self-antigen and we have self-reactive CD8 T cells, right, then these self-reactive CD8 T cells can interact with this, right, because they're good CD8 T cells bind to MHC1. And then we have CD8 that binds MHC1 and the TCR that's binding the self-antigen. Then we know we have our second interaction, which is the B7 and CD28 interaction. And then that's all it needs, right? And then you have activation of these self-reactive CD8 T cells, and they go and then they destroy your beta cells, right? Because these are self-reactive, let's say, against your beta cells, because it escapes central tolerance in your thymus. So that's one way your beta cells can get destroyed in type 1 diabetes mellitus. But let's see what happens here. What if your MHC2 is representing viral antigens, right? Due to the normal phagocytosis process. Well, MHC2 binds CD4 T cells. So we're going to have CD4 binding MHC2, TCR binding viral antigen, and then we're going to have our B7 and CD28 interaction. This will activate the naive CD4 T cell. Macrophages will release cytokines like interleukin 4. If interleukin 4 is released, this naive CD4 T cell, right, can get converted to Th2 and then release interleukin 4 and 5 and therefore activate B cells. So then B cells can class switch and make antibodies, right? This is a normal activation of T cells, right? This is what we want if we have a pathogen. This interaction right here, so we can activate our adaptive immune system, right? So this is just a normal interaction. This is not, causing autoimmunity. So again, if our macrophages release interleukin 12, then the naive CD4 T cell can differentiate into a Th1, which can release, interferon gamma, which attracts and activates macrophages, as we know. And Th1 can release interleukin 2 also, which we know that it can stimulate CD8 T cells. And guess what? If there's self-reactive CD8 T cells nearby, like we just saw here, then this interleukin 2 will stimulate those cells too, and you'll further increase destruction of self-tissue, right? But let's say your antigen-presenting cell took in, right, self-antigen, broke it down via phagolysosome formation, and presented that on MHC2. Then you have naive CD4 cells, but self-reactive naive CD4 cells that recognize the self-antigen. And then the same process occurs here, right? So if this is self-antigen, you, instead of saying naive CD4 T cells, we would say self-reactive naive CD4 T cells, right? would interact with this. And then same thing, all these interactions, same thing would happen here, and you'd get the activation of your self-reactive naive CD4 T cells, right? And then interleukin 4 would be released by the antigen-presenting cell, which will lead to self-reactive Th2 cells, and the interleukin 12 is released by your antigen-presenting cells, right? If interleukin 12 is released, you can get self-reactive Th1 cells. And imagine if these self-reactive Th1 cells are releasing a lot of interferon gamma, you're attracting a lot of macrophages to that area, in this case, your beta cells, right? Your pancreatic tissue. And if these self-reactive Th1 cells release a lot of interleukin 2, again, you're stimulating a lot of self-reactive CD8 cells in the vicinity, okay? So you can see how all of this can lead to destruction of your own tissues, right? And remember, when all of this is happening, your macrophages are releasing a lot of cytokines. If there's a bystander T cell, right, over here, that's not even involved in all of this, right, all this happening, it can get activated and then go to your self tissues and destroy it. So that's bystander activation, right? So let's say you do have a self-reactive CD8 T cell. How is it going to destroy your beta cells? We know that it releases perforins and granzymes, and then it causes beta cell destruction. Okay? But why is this bad? Because the more tissue you destroy, the more damage associated molecular patterns you release, right? The more tissue trauma, which means more activation of your nearby self-reactive T cells, right? Which will again lead to more damage, which will again lead to more autoimmunity, and it's just a whole cycle. Okay, so that's how you get type 1 diabetes mellitus. It's an autoimmune destruction of your T cells. Autoimmune destruction of your beta cells, right? Caused by your T cells, self-reactive T cells. And then imagine if you have no beta cells, then you can't produce insulin. So you would need exogenous insulin, right? So you can manage your blood glucose levels properly. Okay, so that's the overall way and how the bystander effect takes place, and mostly how autoimmune destruction takes place when talking about type 1 diabetes mellitus. All right, hope this video wasn't too confusing. Hope it made a little bit of sense. Hope you guys learned something. Thank you guys for watching, and see you guys in my future videos.